Um, my name is Paloma Contreras, and I am a PhD candidate in biological anthropology here at the University of Michigan. On behalf of everyone in the program, I would like to welcome you all to the last seminar of the Evolution and Human Adaptation Program Colloquium Series. Throughout this semester, we have hosted talented scholars whose work is connected to the topics of genetics, evolution, and human behavior. The talks for this semester were very unique, as the entire series was planned, organized, and executed by graduate students in the Department of Anthropology at the University of Michigan. I want to use this opportunity to thank Miranda Kosman and Charmy Sen for all the work you have put into making this speaker series successful. Before I introduce our guest speaker, I just want to go through some of the technical details for this talk. First, you should see a Q&A icon at the top or the bottom of your screen. You can click this button at any time during the talk to submit your questions, with the option of submitting anonymously. At the end of the talk, we will devote around 10 or 15 minutes to respond to questions. We will not interrupt the talk to ask questions, but rather we'll save all the questions for the end. Second, this talk will be recorded, and I will later post links to the talk on the EHAP Twitter account, EHAP website, and the Department of Anthropology website. Lastly, we would like to thank Julie Winningen for taking care of all the Zoom technicalities and helping us so much with the organization of these talks throughout the semester. With that said, I will proceed to present our guest speaker, Catalina Fernández. First of all, I would like to congratulate her for successfully defending her doctoral dissertation last month. Dr. Fernández just graduated from Indiana University, Bloomington, and subsequently moved to Leipzig, Germany, where she is currently working as a postdoctoral researcher at the Max Planck Institute for Evolutionary Anthropology. Dr. Fernández's work has always emphasized the connections between human biological variation and health outcomes. She started her career at the University of Chile, where she studied traits such as lactase persistence and dairy intake in indigenous populations from the south of Chile. She then expanded her interest to study nutritional transition and health outcomes, food sovereignty, and genetic adaptations to different food products. Dr. Fernández's work has been published in recognized journals such as the American Journal of Human Biology, Current Developments in Nutrition, the American Journal of Physical Anthropology, among others. Throughout her career, Catalina's contribution has not been limited to advancing science. She has also advocated for having more women in science, and she has mentored undergraduate and graduate students from multiple communities underrepresented in academia, including myself. Her research and advocacy has earned recognition from organizations such as the Venner Grant Foundation, NSF, and the American Association of Anthropological Genetics. Today, Dr. Fernández will be presenting a talk titled The Evolutionary Role of Dietary Adaptations and Its Health Outcomes Among Indigenous Populations of Chile. Catalina, thank you so much for being with us today.
Hello, thank you very much, Paloma, for such a nice introduction. And good afternoon, everybody. Thanks for joining me in this talk today. I would like, however, to start also thanking again to Paloma, Charmy, Miranda, Julie, and all the organizers of the EHAP Fall 2020 series. Thank you so much for giving me the opportunity to present my research here today. But none of the work I will present today would have been possible without the people in the Atacameño, Mapuche, and Pehuenche communities where I have worked. I am deeply grateful to them, not only for allowing me in their communities and let me collect their information, but also for their time, conversations, and all the affection during the months I spent there. I also want to thank my research advisors, in particularly to my PhD advisor, Andrea Wiley, and also to Heather Norton, who contributed tremendously to the development of this project, and also to my incipient career as an anthropologist and as a scientist. I want to use this opportunity as well to thank the members of the GeneScape Lab and their PI, Mansa Raghavan, at the University of Chicago, for allowing me in their lab during the many weeks I spent there trying to learn how to analyze my genomic data. In particularly, I have to thank Constanza de la Fuente in the picture for her patience, time, and friendship during that process. And finally, I want to thank my wonderful research assistants in the field in these pictures, Camila Muñoz, Daniela Cordero, Francisca Dahme, and Daniela Castillo, who helped me out during the data collection in the Araucanía region, and also to my research assistants in the Atacama Desert fieldwork, Rayena Aguilar, Paula Candia, Josefa Guerra, and Marcela Paz, who were all always professional and amazing researchers. And of course, I have to acknowledge the institutions that funded my research, the Becas Chile program, the SCOMP Research Feasibility Grant, and the Center for Latin American and Caribbean Studies at Indiana University. And also to the National Science Foundation and Werner Grain Foundation for funding my main research fieldwork. All right, without further ado, today, I would like to talk about how my research integrates the principles of human adaptation to develop hypotheses about the role of nutritional histories in shaping the genetic and physiological characteristics of a population, but also how the interplay between these factors may relate to health outcomes among contemporary groups. So, viewing this way, the research agenda I am developing comprises three main lines of investigation. First, I make use of population genetic approaches and tools to characterize the population structure and demographics that underlie selection events at the genomic level. But more than the population history itself, I am more interested, and I have focused on this very recently, in identifying selection signatures that may be related to environmental and dietary adaptations among populations in South America. Second, I aim to characterize food patterns and understand the drivers of dietary changes in a population. For this, I employ methods from the field of nutritional epidemiology and also ethnographic methods to reconstruct recent changes in the diet. But in addition to that, I also integrate evidence from the archaeological and bioarchaeological records to get a deeper resolution and understanding of dietary changes at a microevolutionary scale. And the third component of my research relates to the interplay between dietary adaptations and health, particularly in changing environments, such as is the case of populations that differ from the Western standard. For this reason, I am particularly interested in understanding how variation in socioeconomic and environmental conditions, needs, genetics, and bodies relates to health outcomes. As you might already know, most of the knowledge we have about the role of genetics, behavior, environment, etc. in health, and also about how we even define health in general, has been constructed based on people of European descent and living in contemporary urban industrialized environments. These populations have been described as weird, like Western, educated, industrialized, rich, and democratic, but also weird in the sense that they only represent a very minimal fraction of the world population. Yet, they correspond to the vast majority of study participants in almost every field that deals with human biology and behavior. In my research, I have focused on non-weird populations, and in particularly, I have worked with indigenous communities of the north and south of Chile, because there is where I have years of experience and also where I have developed trust with the communities with whom I work. So early on in my career, actually as an undergrad in Chile, I became interested in studying the consequences of particular foods that became global and also normative in diverse cultural and biological contexts. So the case of lactase non-persistent and food policies that promote or even enforce dairy consumption among populations who don't even have a milking history seems like an interesting research problem. So motivated by this idea, I conducted fieldwork research where I took DNA samples and I applied several questionnaires to characterize the genetic pattern of lactase non-persistence among the Mapuche and to see if there wasn't any relationship with dietary patterns and symptoms related to lactose intolerance. So what I found is that compared to other populations in the country, they had the lowest genotype frequencies of lactase persistence, so about like 10%, and also had the lowest consumption of milk and dairy. However, contrary to what I was expecting, individuals reported not experiencing strong symptoms of lactose intolerance when consuming milk, although they rarely have large quantities. So that might also explain in part why. But besides, from their perspective and contrasting, to other populations where milk is not common, they actually value milk and dairy, but more as a food to enhance children growth and not something they are used to use for like cooking. So along the same lines, and because I continue to be interested in the role of food and nutrition in adaptation and human evolution, I have also conducted research on the second most popular example of dietary adaptations, which is copy number variation at the salivary amylase gene among humans populations. So in this research, I systematized all the studies assessing copy number variation in contemporary populations published by the beginning of 2017. And what I found is that there's actually quite a big range of variation, even in populations that are deemed as contrasting in terms of their dietary history of starch consumption, such as the Hadza and the Mbure, which are here in green and purple dots. So in your left, like the ones in green are the high starch diets, and the lower starch diet are in purple. In fact, most of the populations that were sampled, have around 6 and 7 salivary amylase gene copies despite their diet. So this partially kind of challenge the hypothesis that amylase copy number variation relates to starch consumption or history of this diet. So although I do not have a conclusive explanation of why salivary amylase varies so much in humans, I found that diet might not be the main driver of this variation. But since I do not have enough time to go into detail about this, we can discuss it more at the end, or you can also take a look at our article we published in American Journal of Physical Anthropology. So building on these two examples that I just mentioned, that study food and diet from an evolutionary and ecological perspective, for my dissertation, I conceptualized a research project in which I integrated nutritional histories, genomic tools, and nutritional epidemiology methods to understand whether relatively recent dietary shifts had evolutionary consequences for our species. And also I asked, to what extent these are related to the rise in chronic diseases among contemporary populations? From an anthropological and also an evolutionary perspective, this is a major puzzle that motivated me to ask the main research question in my study. So, which is, as you can read here, I was interested in understanding how differences in past subsistence strategies, in this case, foragers versus agropastoralists, may have led to different genetic adaptations in genes that are related to metabolism, and how those adaptations may relate to modern-day well-being and chronic conditions. To do so, I have conducted fieldwork research in the Araucanía region, which is highlighted in light blue in the map on your right. This region harbors a numerous indigenous population from the Pehuenche ethnic group here in the west side, which is close to the border with Argentina. On the other hand, the part of the north of Chile, highlighted in dark blue and close to the border with Bolivia and Argentina, it's occupied by the Atacameño or Licanantay ethnic group. As you can see, these are highly contrasting natural environments. The region occupied by the Pehuenche in the south maps to temperate rainforest, and although it's in the Andes, it's not a high altitude zone. In contrast, the region inhabited by the Atacameños correspond to the Atacama Desert, and it's characterized by its extreme aridity, altitudes that range from 2 to over 4,000 meters above the sea level, and also the few available water sources they have are very high in arsenic. In the region occupied by the Atacameños, due to the harsh conditions of the Atacama Desert, villages are located in the few areas that have some access to water, which are the Atacama Desert oases and also the Loa River ravines. In the map on your right, the oases are here surrounding the Atacama Salt Flat, that white mass, and the Loa River ravines are in this northern part in the map. I have also indicated in black and green dots the communities I sampled for this research. And as other communities, populations in the Andes, the Atacameños have a long tradition of agriculture and pastoralism that date back to over 3,000 to 4,000 years. We also know that until a few decades ago, they dedicated mainly to llama pastoralism and agriculture of corn, potatoes, and quinoa, among other species that were introduced with Spanish colonization. However, this has changed dramatically in the last few decades due to the expansion of copper mining, and also lithium, mainly lithium extraction in the region. As a reference on your right, you can see a picture of one of the biggest oases in the community, and one of also the biggest community in the region, which is Toconao. And although this is one of the most populated communities, you can see from the scale below that it's quite small. The other population where I have conducted research is the Pehuenche, which are a Mapudungun speaking and Mapuche subgroup. The area occupied by the Pehuenche used to be densely covered by forest, but deforestation has accelerated in the last few decades, and in many places, native forests have been replaced by pine trees due to the mainly incentives to the logging industry in the region. So compared to the Atacameños, we don't know as much about the food history of this population before colonization, but we do know from several very early Spanish chronicles that agriculture was not practiced in this specific area. Instead, people in this region based their subsistence on foraging of several wild fruits and mushrooms, many of which are still part of their diet. The main ones are the pehuen seeds, which are here in the middle in the picture, which are the fruits of the Araucaria araucana, a tree that is all over the higher altitudes in this region. All right, so in these two field sites and populations, I collected a rich and unique set of data on food change and nutrition transition through participant observation, semi-structured interviews, and a scale-based questionnaire I designed to quantify the nutrition transition to a Western diet. I also collected detailed data on current food consumption using a food frequency questionnaire and patterns of physical activity using a version of the WHO questionnaire, which I adapted to the local context. Then, with the help of my wonderful research assistants, I collected data on several biomarkers of non-transmissible chronic diseases, such as systolic and diastolic blood pressures, and then anthropometric measurements such as height, weight, body fat percentage, and waist circumference. For other biomarkers of chronic diseases, I collected capillary blood samples to measure blood glucose, total cholesterol, HDL cholesterol, and triglycerides. And finally, I collected DNA samples from saliva, for which I then extracted the DNA in Mauricio Moraga's lab in the Universidad de Chile. So thank you also to Mauricio as well for his help and facilitating the space and equipment. I processed the samples there, and then I sent these samples to an array genotyping facility to genotype over 800,000 markers across the genome using the Axiom LAT, which resulted in a genomic data set for 177 individuals. I collected most of the data I just described during the 8 months of fieldwork I conducted between 2017 and 18. But preliminary fieldwork I did over the summers of 2015 and 16 were crucial for this research, as it allows me not only to better frame my questions and fieldwork logistics, but also, most importantly, to meet with community leaders and members to discuss this project with them. At each community, I recruited participants that were older than 18 years old and who also self-identify as belonging to one of these two ethnic groups, and also who reported at least 2 previous generations of relatives in these communities. As exclusion criteria, I avoided pregnant women and individuals that were highly related by kinship. So the main objective of this part of my research was to identify signatures of selection and candidates' needs in genomic regions involving nutrient digestion and metabolism among these 2 distinct populations, which have a long histories of different subsistence strategies. As you may or may not remember, the Pehuenche, a history of foraging, and the Atacameños, a history of agropastoralism. So first, in order to characterize the genetic diversity of my datasets and evaluate how this diversity is organized, I ran a principal component analysis, including a subset of African, Asian, and European samples, and all individuals from Latin America in the 1,000 genomes database. And as you can see here in this light brown on the top left, we can identify a cluster with individuals from Africa, then in the bottom, individuals from Iberian Peninsula, and the cluster in gray correspond to East Asians. So the individuals circled in here correspond to my samples plus individuals from other indigenous communities in South America. I mean, a magnification of this region, the region I circled in the previous slide, allow us to better distinguish the close relationships between the American samples included in this study. However, from this plot, it's not possible to clearly distinguish population from the north, like the Aymara and the Atacameños, which are in yellow and green in these plots, from the Mapuche subgroups in the south, which are the blue crosses and pink and purple dots in here. So to further look at the genetic structure of my dataset, I combine it with a reference panel from populations outside the Americas and also contemporary admixed populations in this region. This analysis allows the characterization of a number of ancestral groups and the proportion of these groups in each individual. So in this plot, I am displaying a total of 6 ancestral components, which was the number of groups that better describe this data. Each column represents an individual, and each color is a different ancestral component. Circled in red and blue in the bottom right are the 2 populations sampled for the study, and it is clear that populations from the Mapuche subgroups, including the Pehuenches, share a genetic component here in orange. On the other hand, a component clearly discriminates between the Atacameños in red and Aymara in blue, which is another indigenous population in the Andes region. I also estimated local ancestries using RFMEX, which identify at the individual and chromosome level the portions of each chromosome that trace ancestry to predefined continental groups. So in here, African ancestries are represented in red, European in blue, and American in green. And this is just an example of 2 individuals of my dataset. The one on the right corresponds to a relatively admixed Atacameño who has consistent and relatively high African and European ancestries. And in contrast, in the left, there is an individual that has very low admixture with other continental groups. So then, using a subset of individuals with over 99% American ancestries, I ran these 3 different selection analyses. First, the population branch statistic, abbreviated as PBS, which is a frequency-based method, and then 2 haplotype-based methods, the integrated haplotype statistic, abbreviated as IHS, and the cross-population extended haplotype homozygosity, abbreviated as XPIHH. Among the main results of the PBS analysis between the Atacameño, Pehuenche, and East Asians were the high scores in the PAKB4 gene that encodes a key enzyme in glycolysis. Then in chromosome 10, I found consistent high scores in the gene AS3MT that encodes an enzyme that metabolizes arsenic. And in chromosome 15, I found a signature in the FBN1 gene, which encodes for 2 proteins, profibrillin and also astrosine, which is a fasting-induced glucogenic hormone. In the HHS analysis, among the Pehuenche, I found a consistent signature in chromosome 6, corresponding to one of the HLA genes involved in immune function. And other consistent high scores were observed in chromosome 6, 7, and 13, but I will not go into detail about this for now. Regarding IHS scores among the Atacameños, I found again a signature in the HLA complex, which relates to the immune function. So then consistently, I found high scores in the ACACB gene in chromosome 12, which encodes a crucial enzyme in the process of fatty acid metabolism. And in chromosome 20, high scores in the CYP24 that codes for enzymes involving vitamin D and calcium metabolism. And finally, the results of the cross-population EHH between the Atacameño and Pehuenche show high scores in chromosome 7, mapping to these 2 genes in here, one of which encodes for an enzyme involving adipogenesis. Then in chromosome 10, we have the gene LRMDA that encodes a protein required for melanocyte differentiation, and TYR, which encodes for an enzyme that catalyzes melanin production. I finally ran an enrichment analysis in the top 5% windows for all analyses to identify genes that are overrepresented in the top scores. The most significant results were in the cross-population EHH analysis between Atacameños and Pehuenche. For this one, I found that the genes that belong to the response to nutrient pathway were significantly overrepresented in this 5%. And in particularly, And this included genes in the glucose and fat metabolism, and also in the vitamin D and calcium metabolism. So to summarize the main findings of this part of my research, I identify a clear structure among South American indigenous populations, which is consistent with major geographical regions, particularly the Andes and the lowlands. And regarding selection analysis, I found a consistent selection signature in first an enzyme that metabolized arsenic among the Tacameños, which makes sense. Then signatures and genes related to skin pigmentation, and also signatures related to glucose and fat metabolism, which in several cases are consistent with a response to a prolonged fasting. So regarding the second component of the research agenda I am developing, which relates specifically to population nutritional patterns and their transformations through time, I use a socioecological and evolutionary approach to understand the main drivers and consequences of these changes among the Atacameño and Pehuenche communities I study for my dissertation. For this particular research, I characterized the dietary and physical activity patterns and the agents, also the agents causing transformations in the lifestyle and food systems at these two populations as reported by the individuals. So for this portion of the study, I analyzed data from participant observation, field notes, and semi-structured interviews, which I used to provide an historical and cultural context to my findings. I also analyzed nutritional and physical activity data I collected through questionnaires. Among the main results of this part of my research, I found that agriculture and pastoralist traditions, and also knowledge, are being lost and at a very accelerated rate, mainly in the last 2 generations. In fact, today are mostly the elders of the communities who are in charge of the family agriculture labor and knowledge, and very likely these communities might abandon this practice in the next generation, as these experiences are not being transmitted to the young. So more on this process is in this book chapter on family agriculture among the Atacameños that I co-author with my research assistants. Now, when I compare some of the health and also food-related health patterns between the Pehuenche and Atacameño samples, I found a few interesting things. So I know there is a lot in here, but just look at the highlighted lines. So, well, first, in the Atacameño sample was significantly older than the Pehuenche, and consistently physical activity was higher among the Pehuenche. Regarding some dietary patterns of interest, the proportion of ultra-processed foods in their diet, but also traditional foods, as defined by the participants, were higher among the Pehuenches. On the other hand, and consistent in the 3 questions about individual and parents' food insecurity, food insecurity was significantly higher among the Pehuenche. The main findings of this part of my research are first, well, there is a trend of lower consumption of local foods or traditional foods over time, and many local foods that gather or produce are currently directed to informal markets, so families can purchase other less nutritious foods, but that are seen as more essential, such as sugar, vegetable oil, white flour, and rice. Second, I found that despite contrasting ecological and historical contexts, the 2 groups are converging into similar process of market and wage labor integration. And although some aspects of their traditional foodways are still in practice, overall, they're transitioning to a Western diet high in processed and non-local foods. Now, because the ultimate goal of my research agenda is to understand whether dietary adaptations are related to health in non-weird contemporary populations, I investigated whether differences in past subsistence strategies or dietary adaptations may contribute to explain differences in some health outcomes. And second, in order to integrate the biomarker data and the selection scans, I estimated the association between biomarkers of chronic diseases risk and also a list of candidate SNPs involving nutrient transport and metabolism in these 2 populations. So for the first part, I was mainly interested in testing whether ethnicity, but actually it would be more accurate to say a history of agropastoralism or foraging, played any role in chronic disease risk. So after categorizing each of the biomarkers values at risk or not at risk, according to the NIH cutoffs, I found that the likelihood of being at risk for any of the markers I evaluated, such as high blood pressure, high cholesterol, etc., did not differ between the Atacameños and Pehuenche. In these models, of course, I control for the effect of age, sex, food insecurity, ultra-processed foods, and physical activity. In contrast, but as seen in most populations, the variables that were more frequently associated with risk across the health outcomes that I evaluated were age and sex. I also use several of the biomarkers I measure plus other several risk factors such as smoking to calculate Framingham risk score, which is a sex-specific algorithm to estimate cardiovascular risk. And again, I found that only age and sex were significant predictors of this risk. The main finding regarding health outcomes, well, I found no significant differences in chronic diseases between the two populations, which is consistent with the relatively similar degree of participation in the wage labor and market integration that have both uniformly affected their food system and lifestyle patterns. And overall, chronic disease risk is relatively high at both populations and resembles the trend I observed in other indigenous populations that have gone through nutrition transition. So then, in order to put together these health-related biomarkers and the genomic scan, I selected the SNPs that were significant in the enrichment analysis for the response to nutrient, and also the positions in the top 1% of the selection analysis that were involved in glucose or fat metabolism. So this resulted in 31 SNPs that I used to run some association analysis with health outcomes. So the only consistent patterns I found were among the Pehuenche for the gene FBN1 and height. Remember that this gene was the one that encodes profibrillin, but it's also a fasting-induced glucogenic hormone. So this gene was already associated with height in a GWAS conducted in a Peruvian population. And the authors of that study explained that pattern as an adaptation to the coastal environment. However, this hypothesis does not hold for the populations studied here because none of them are coastal populations. The other pattern I found was for the ACSL1 gene among the Atacameños, which encodes an enzyme involving fatty acid metabolism and triglyceride synthesis as well. And variation of this gene was found to be associated with BMI, body fat percentage, and HDL cholesterol in this population, in the Atacameños. So overall, this analysis found only a slight support for the role of past subsistence strategies in current NCD risk, but this association between selected SNPs and chronic disease risk was found for the former agropastoralists, the Atacameños, and not in the forager population, as it would have been predicted by the evolutionary mismatch hypothesis between past and current environments. As an alternative explanation, and considering the function of the genes selected among the Atacameños, it might be that this signature is driven by seasonal famines in the past, but definitely additional archaeological and also bioarchaeological data is needed to test this hypothesis. On the other hand, and as a final thought, I would like to emphasize that the phenotypes I study and I show here are very complex, complex phenotypes. And unlike the lactase persistent example that inspired me very early in my career, it will probably be very unlikely to find any SNPs that explain a biologically meaningful portion of the variation in these health outcomes. So as for the short-term directions of this specific project, I'm working on integrating population structure data on the association analysis. So this should definitely strengthen or rule out some of the associations I observe. And regarding additional work that I would like to do using these genomic data, I plan to examine in more detail the demographic history of the Atacama Desert, in particularly to disentangle the African ancestries I found in the region and also the timing of admixture. Regarding the selection analysis, I plan to run additional selection tests particularly tests that allow me to better distinguish between signatures of recent selection versus signatures that occur probably before the populations in the Americas split. And furthermore, I am also systematizing the data on the interviews I collected to discuss the connection between food and memory, and on the conflicting narratives on past food insecurity, health, and progress, as this was a theme, definitely, that emerged very frequently at my interviews. And in general, I plan to continue my work on studying food and health from an anthropological and evolutionary perspective among populations that have been historically marginalized from these studies, so non-weird populations. Yeah, so this is pretty much what I wanted to share with you today. Thank you very much for joining me, and I'm happy to take any questions you may have.
Thank you so much for your presentation, Catalina. This was amazing. I want to remind everyone that if you have any questions or comments, you can click the Q&A icon at the top or the bottom of your screen and type them there, and I'm gonna read them for you. So I have a few technical questions. So I would like to know how you defined food insecurity. And also I'm curious about the signals that you found related to vitamin D and calcium, and if you have any hypothesis to explain that.
Sorry, I missed the first part of your question. I got the part of calcium and vitamin D, but I missed the other part, sorry.
No worries. The first question is a very technical one, was how did you define food insecurity?
How I ask for food insecurity?
Yeah, or how will you conceptualize it? How do you measure it?
How do I what? Sorry.
How do you measure it?
Oh, how do I measure it? Sorry. Yeah, so regarding the question about food insecurity, I basically apply a questionnaire that is used to evaluate food insecurity. I ask individuals about experiences of hunger in the past, especially during their childhood. And the reason I did that is because I wanted to have some sort of way to measure variables that are related to the development, origins of health and disease hypothesis. So to see if there was any relationship between maybe individuals that suffer from food insecurity during childhood and health outcomes, negative health outcomes, such as especially diabetes, since this association has been observed before. I wanted to also have data to better measure this data on birth weight, which is a really good way to actually capture early exposures to food insecurity. But of course, most of the populations that I work with were actually not born in hospitals, so they did not have a record of how much they weigh. And so that was a way to disentangle any role of the variables associated with this the DAAS hypothesis. And regarding your question on the signatures on vitamin D and calcium, I think this is something that I'm still exploring. I just finished my PhD, and this is not something that I have published yet. I think that some of the signatures might be related with pigmentation as well. So I found a couple of genes that are associated with more than pigmentation itself, like tanning ability. That it's been observed that it's associated with tanning ability. So it might be that there's some sort of relationship there, especially considering that some of these populations are exposed to the sun a lot, and especially in the Atacama Desert. So some sort of protection needs to be in place. So it might be something related to that. That's what I think. But this is just a thought, not something that I have worked out very well yet.
Mm-hmm.
Thanks.
So we have one additional question about pigmentation, actually. So the question says, your research is really interesting. Thanks for the great presentation. Were the pigmentation selection signals, such as TYR, both in the Atacameño and Pehuenche populations?
No, so this I found it in the Atacameño population. Yeah, in the Pehuenche only I found signatures related to only to immune function. I mean, I found others that were not really biologically easy to interpret. So since I mean a lot of signatures that you find in this test are actually related to cellular processes. So it's really hard to actually make sense of the signatures. So I basically presented the ones that have more clear phenotypes. Yeah, so.
Mhm. The next question is by Maria Nieves. She says, brutal tu presentación, Catalina. Felicidades. And she's asking, do you know if any of the candidate selection genes you identified have also been reported as potential selection targets in other Latin American or indigenous populations?
Yeah, as I mentioned, the FBN1 gene, which I found a strong signature in the Pehuenche, is associated with height in my sample, and it's also been shown to be associated with height in coastal populations in Peru. They also conducted some selection analysis, and they found that region is under selection, apparently, but there's no really clear explanation of why. And they say, I mean, their explanation is that it's an adaptation to a coastal environment. But honestly, I don't really understand how height might be related to maybe an adaptation to a coastal environment. And it's usually thought that height is related to, for instance, living in a tropical area. That's something that's being studied for a while. But we actually have very different hypotheses of why, but not really about what would be the signature behind of this. Yeah.
Mm-hmm. The next question says, thank you, Catalina. Do you think your results will remain the same if you added more populations, possibly Patagonian populations?
Remain the same. Like, you mean? Sorry, I think I need more clarification when the anonymous attendee.
I something to add to that question, because I was wondering too if there is any way to distinguish the selection signals that you observe as something that happened before these populations arrived to those environments or after. Do you know anything about the demographic history of migration in these populations?
So yeah, so that's something that I said as like the future, not that like short term, but future direction. So something that I actually should start doing like soon. It's like actually run other tests of selection that are better at distinguishing more ancient signatures. So for instance, I start already running instead of the PBS, an analysis that's called the ABS, which is ancestral branch statistics. So to know if like there's a signature maybe that is common to these populations. So they both have it, and it's actually something that could have happened before the populations entered the Americas. So not necessarily a population-specific signature. Yeah, I'm trying to think more about like the question on incorporating Patagonian populations. I don't know exactly where that question go. If do you have any, or can you please add more on the question, elaborate more?
We'll see if this anonymous person can elaborate a little bit more on that question. I have one additional question that maybe is connected to the previous one. I wonder if you saw any signals of natural selection connected to high altitude adaptation?
No, I didn't find anything related to high altitude. And something else that I also found, it's actually an answer to Maria's question, is I found a signature in the same gene that has also been observed under selection, or whose like genotype frequencies are very high in populations that live in the same region, but not actually in the Atacama. In the Atacameño, they're not from the Atacameños, but they are living in very similar environment, whose water sources are very high in arsenic as well. So in in Argentina and also in the north of Chile, northern Andes, as is being found also a signature on the AS3MT gene, the arsenic metabolizing gene. So, and I found that as well. So that is was kind of a sanity check for me, like finding this in the Atacameños and not in the other, and actually finding this. So yeah, it means that at least the other results that I am observing have some sort of validation. Yeah.
Mm-hmm. So we don't have any other questions, but now, give me a second. I would like to ask Miranda to intervene. Before we end, one of the co-organizers is going to close out this series for this semester. So Miranda, do you want to jump in?
Yeah, hi. So first of all, thank you, Catalina, so much for a really wonderful talk and a really interesting talk. And again, we are so grateful that we were able to have you come and present. So I would also like to thank everyone for your attendance throughout this series. My co-organizer, Sharmi, and I would like to thank a few people just really quickly. So first, we'd like to thank Julie Winningham for her help and facilitation of the Zoom webinar. We would also like to thank our two faculty who helped us with organizing the series. And a huge thank you to all of our our fellow graduate student hosts who did an awesome job, like Paloma. And finally, we would like to thank all of the speakers who participated. We are really grateful that you took time out of a very hectic and a very difficult semester to participate in this symposium. This is the last seminar of the semester. So again, thank you to everyone who attended, and we hope that the people listening to us now can join us for more speaker series in the future. Watch the EHAP Twitter and our website for more details. So thank you, everyone. Have a great rest of your day and happy holidays.
Thank you.